Hello, my name is Douglas Lin, and this is the recorded presentation for my project on computationally modeling the role of intercellular interactions on single cell and population level responses. So the simulation of cellular behavior can be achieved through kinetic modeling, where we basically understand how cells behave over time. And this is an active field of research in systems biology and biophysics especially. However, the dynamics of multicellular systems have not been well studied, and this is partially because of the establishment of many single cell modeling tools, however, the lack of multicellular modeling tools. And this is a shame because there's great potential for leveraging natural phenomenon involving cellular interactions, such as bacterial quorum sensing, for the design and use of multicellular biosensors, which could serve to amplify the signal. So in this work, I constructed an interoperable framework for multicellular modeling that would be compatible with existing frameworks such as virtual cell to increase the ease of use. And I used this system to answer the research question of how intercellular signaling affects unicellular and population level responses in a positive feedback loop, as you might see in a biosensor. So I simulated one cell and then compared its behavior over time to systems of 10, 25, 50, 75, or 100 cells. So chemical kinetics govern how reactions proceed with respect to time. And the law of mass action states that the rate of a reaction is directly proportional to the concentration of reactants. So in this project, I used the stochastic Gillespie algorithm to model the chemical kinetics that occurs within cells because it's more accurate on the nanoscale rather than the differential equations that will work on the macro scale. So essentially the way the Gillespie algorithm works is it randomly decides which reaction is the most likely to next occur and then predicts the amount of time it takes for that reaction to occur. And then it basically repeats this process until the simulation time is met. So this is design of my software. So first you design the biochemical network in virtual cell because it has a very nice user interface. And then what my program does is it extracts the intracellular and extracellular environments and then duplicates the intracellular environment so that they all share the same extracellular environment. It then simulates this system and then you get different cellular trajectories which you can aggregate for analysis. So this is the biochemical system that I simulated to test my hypothesis. Essentially, this is multiple cells that they all produce a cell membrane permeable transcription factor. I call it TF, and this transcription factor stimulates the production of a reporter protein. So these are the graphs of the amount of reporter protein produced by each cell over time. So the inset graphs represent the cellular trajectories for a single trial, and then the larger one represents the cellular trajectories over 50 trials, averaged over 50 trials. So here are the plots of the amount of reporter protein present at the end of simulation time in each cell and in total in each system. The amount of reporter protein increased as the amount of cells simulated did in general. However, at 20 seconds of simulation time, the amount of reporter produced per cell actually decreases as more cells are simulated. This could be because the amount of intercellular transcription factor is diluted amongst more cells, and this results in a delayed response. But by 50 seconds, this effect is counteracted by the increasing number of cells producing the reporter and is therefore much higher. So given these results, I modeled a proof of concept multicellular bioreporter design that has two shared intercellular species that trigger the production of a reporter protein, which is what's plotted on the right. So the analyte will be the contaminant that it is supposed to measure, and then the signal is a messenger protein that diffuses across cell walls and is used to basically amplify the signal and trigger other cells to produce more of the reporter protein. So as shown, more bioreporters in a system produce a greater response on the single and multicellular level, indicating that it could be used for the sensitive detection of contaminants. So to recap, my simulator was able to recreate the core functionality of virtual cell while also extending its capabilities to multiple cells and having finer control over its implementation details. And future work would include the simulation of heterogeneous cell populations and the simulation and validation of real multicellular bioreporter designs with real rate constants derived from experimental procedures. So in conclusion, I successfully constructed an interoperable multicellular stochastic simulator and found statistically significant differences in cellular and system level responses when intercellular interactions were accounted for. I also demonstrated the nuance of simulating multiple cells by showing different behaviors at different simulation steps. And this all shows a promising area of development for multicellular bioreporter design and simulation. And it could be used for the enhanced detection of environmental contaminants such as PFAS, heavy metals, PCB, and other hard to detect contaminants like those. So I'd like to thank Dr. Michael Martin of the University of Louisville Micro and Nanotechnology Center for his inspiration and guidance, and also my iGEM team who inspired the whole project. And thank you for listening.